can turn on. I think this is almost as full of a class as a regular day. All right, let me share my screen. Okay, I did. Oh, I need iClicker. Okay. We'll start that. It feels like it moves very quickly and very slowly all at the same time. I will agree with that. Okay, so y'all, attendance is open. You can go ahead and check in on attendance. Very quickly and very slowly all at the same time. All right, I want our class. And I didn't bring any handouts of the long iClicker because it was a virtual day, so I didn't think I'd see very many of you, but it's okay. Y'all have it on, have access to it on Moodle, right? Okay. All right. So we got 3 more minutes. No, it's going to be like the longest section today. Very short, very short. We're focusing on the receptors that you're going to need for like glycolysis and that you're going to need for the next ones instead of on all of them. So there won't be too much detail, just a good overview for this last section, kind of like we did on the previous section. Shortest video, right?
10 minutes.
10 minutes. That was it. 10 minutes. Y'all got 10 minutes. All right, y'all. Want y'all joke?
Go for it.
It's really cheesy today. It's really cheesy today. What do you call a dinosaur that is sleeping? A dino snore. Charles liked that one. Charles actually liked that one. But that's okay. It's okay. All right. Thank you. Yeah, that is a congratulations for me because he usually doesn't laugh at my jokes. All right. So I wanted to tell y'all about one change that I made to the schedule. So if you go to our pre-recorded lecture schedule, right? Come over all the way to the end, right? All the way to the end. We had an exam scheduled for the last Friday of class that was going to cover the last 3 chapters. But I didn't know about your dead days, right? Do y'all even know about these? Yeah, you're not allowed to have tests. I didn't know this. So you're not allowed to have tests. So we're not going to have a test on those days. What I'm going to do is I moved your 4th exam back 1 week. So it's only going to cover 2 chapters. And then we'll cover the 3rd chapter. And then the final exam will be cumulative. So that last chapter, chapter 11, will be on your final exam.
So it's not on the 13th.
It's not on the 13th anymore. It is on the 6th. It is on the 6th.
And it's not for chapters
9 and 10. So I didn't know. So
It's okay, no, honestly, I forgot to.
seems like everybody did. That's okay. All right. So just make sure you change your calendars so you have that. But that'll be kind of nice. It'll be less material on the exam, on that 4th exam as well. The final, yes, the final has not changed. The final is
Still November 18th.
still Wednesday, 16, 17, 18. Yep. From 1 to 3 p.m. Any questions on that? Y'all good? Okay. All right. Awesome. Awesome. And don't forget what is due tomorrow night. Our self-checks for the 2 chapters that are going to be on the exam, chapters 7 and 8. Okay. So make sure you have those done. You have unlimited time and 3 tries. Get them all right.
I did chapter, I got chapters.
Okay.
I didn't complete chapter 7.
All right. So let's come over here and we're going to do lecture on
The deadline for the term is like Tuesday night, right?
Tuesday night because the test is on Wednesday. So I'm giving you as much time as possible. And then as soon as it's all done, I'll release all the answers and stuff. Because you normally get that feedback right away. But now you have to wait till it closes.
Yeah.
Do you get, like, know if you got any wrong?
You do. You do. So as you're taking it,
It just doesn't tell you which one.
it just doesn't tell you which ones. Does it? No, I think it tells you which one you got wrong, but it doesn't tell you the right answer.
Exactly.
Anybody who's taken it?
Yeah, I've taken it.
So it tells you if you got on the self checks, it tells you if everybody wants to push the exam back. It tells you if you got it right or wrong, but it doesn't tell you what the right answer was. So that you know which ones to go back and,
Except for the animations, like if you have to, if it's a discussion question, it doesn't.
yeah, no, the discussion question does not. The discussion question does not. Does not tell you if you got it right or wrong.
I guess that's because like that's open ended.
Y'all, y'all, we cannot push it back. We cannot push it back.
I kind of like the Wednesday.
We cannot push it back.
I'll support you. I already
We have Vine camps, but I'm not pushing it back, so I'm sorry. All right, so let's do this. Edit full. We want to view full screen. I think yes. All right. So nuclear receptor signaling, right? This is not too bad. I think that's part 2. I think I'm back 1. Yeah. Okay. Sorry, y'all. All right. Nuclear receptor signaling. Yes. Yeah. Is it recording? The red dot is recording, right? Pause. Yeah, it's recording. Yes. I'm sorry, y'all. I'm sorry. Okay. So nuclear receptor signaling. Nuclear receptor signaling is the only signaling that occurs where our receptors are not on the outside of the cell. So these are the ones that are known as our intracellular receptors. So they're not bound to the membrane. What do we typically call them?
Membrane bound receptors?
Not membrane bound receptors, non-membrane bound. We call them transcription factors. Transcription factors. Yeah, exactly. You're on the right track. You're good. So what do transcription factors do? They regulate gene expression. So you've had classes where you've talked about that before, right? No? Y'all are just pulling my leg today. Yeah. I see how it is. All right, so they serve as transcription factors. There are two major classes of these nuclear receptors or transcription factors. The words are interchangeable. They could be steroid receptors or they could be metabolite receptors. So when you think about the two, what are steroids derivatives of? Cholesterol. So they're all going to be cholesterol based. They're all going to have that steroid nucleus, so that common structure that all cholesterol derivatives have. And we'll talk about some metabolite examples as well. So there are three things that are going to govern basically how these receptors signal. Number one, every cell type is going to express a different set of nuclear receptors. Like if you're in a kidney cell, do you want to have the transcription factors that transcribe epithelial cell markers? No, so they're not going to be present. How is that controlled? What do we do to DNA that we have to keep, right? Every cell has the complete book. That's what I call it. Every cell has the complete book. But cell types in specific express certain chapters, right? And that's what makes them the specific cell type. So how do we do that? How do we only express one chapter of the book? It's all about condensation of the DNA. So it's all controlled by chromatin, right? So, so that's one thing. Whether you have the right nuclear receptors in the nucleus, you condense the rest, that's right. Localized bioavailability of ligands. That means, is the ligand anywhere near the cell that needs the message, right? If you don't make the ligand, the cell's not going to get the message. So that's a pretty simple one. The last one is differential accessibility of our target gene DNA sequences. So this is very similar to number one, right? Because just because you have the transcription factor for the gene, does that mean that that gene area is decondensed? And not necessarily. It depends on the environmental factors, what parts of the DNA are condensed and what parts are not, right? So you might have the transcription factor, but then there's no binding site for the transcription factor because that bit of DNA is tied up in the chromatin. Does that make sense? All right, so those are our regulatory parameters. Now, here's our two basic, can't go down, there we go, our two basic receptors, right? We have steroid receptors here on the left, and we have metabolite on the right. What you're gonna notice is that there are some commonalities in all steroid receptors. What you can see is that these are homodimers. So do you see how they're the same color, they're the same sort of shape, right? They bind the same ligand. That's a homodimer. And what you'll notice is they're basically mirror images of one another. So what does that tell you about the sequence of DNA that they're going to bind to? If they're mirror images, then they have to bind to inverted DNA sequences, which means that if you have your transcription factor 1 binding to AGAAC, you're going to have that same sequence here, AGAACA. Sorry, I think I left off the A, right? But these are inverted repeat sequences. Okay? Look at our second type. These are our metabolite receptors. Are they the same? No, they're different. So they're hetero, but we still need two of them. Well, I'll put you a caveat. Most of the time it's two of them. Sometimes it's only one. It's usually two. One of them is usually specific for the gene that's being transcribed. And then one of them is usually a common transcription factor called RXR, or the retinoid X receptor. That's just where it was first discovered. But you don't have to know that that's the one it is. You just have to know this one's a homodimer and this one's a heterodimer. And what do you notice about the configuration of the two?
They are like a mirror image of each other. They're one side different from the other.
One side, so this is like a head-to-head interaction, and this is a head-to-tail interaction.
Asymmetrical.
Yes, you could say that, yes. So what you have are direct repeat DNA sequences. So if this one binds to A, G, G, T, C, A, this one binds to A, G, G, T, C, A. Okay. So what kind of ligands are going to bind to steroid receptors? If you have a glucocorticoid receptor, what kind of ligand are you going to have? Glucocorticoid. If you have an estrogen receptor, what's your ligand? Estrogen, right? So on and so on and so on. The only one that's kind of weird is this one, is the thyroid hormone receptor. That's actually a metabolite receptor. But I won't ask you that on the exam, I promise, because that's kind of mean. Okay. All right. Oh, and the other thing I wanted to note is both of these types of transcription factors require other proteins in order to go. And so we call these co-regulatory proteins. So no matter which class you're in, you need those co-regulatory proteins. So no matter which one you are going to have a lipophilic first messenger. What's a lipophilic first messenger? A ligand that can what? Can pass through membrane. Okay. Now what's cool about these transcription factors is that they can do it one of two ways. You can bind the ligand to the transcription factor when the transcription factor is connected to the DNA, like it was in that picture, or the transcription factor can be like loose in the nucleus, bind the ligand, and then go find the DNA. So the binding can occur, the binding of the ligand to the receptor can occur with or without the DNA present, either one. And once that ligand's activated, then it's going to recruit your co-regulatory proteins. So what does all this do? How does this change the DNA? Well, usually, not always, but usually it's through acetylation or deacetylation of chromatin associated proteins. And acetylation and deacetylation, this mechanism is basically an on-off switch for genes. So it changes their packing, it makes them more accessible or less accessible, and so this is how you very quickly turn genes on and off inside of the cell. That's your on-off switch. Okay, so this is what we said before. Steroid receptors are in head-to-head homodimers. We showed that in the picture, that combine to our inverted DNA sequences, right? And we already said that ligands are cholesterol derivatives. And so what's our most common ones? These are hormones, right? Cholesterol derivatives are hormones. Hormones. Mm-hmm. Not all, but the most common. The most common. Okay. So our metabolite receptors are our head to tail. So that's the opposite, right? And because they're head to tail, they're going to bind to the exact same DNA sequence. And you do not need to memorize the DNA sequence that they bind to. Okay? Don't do that. Good? Don't do that. Ligands. Ligands can come basically from dietary nutrients. D-I-E-T-A-R-Y, dietary?
Yeah.
Thank you. Dietary nutrients. So things that we consume, like vitamins, like unsaturated fatty acids, essential amino acids, right? Like vitamin D, like retinoic acid, those kinds of things, right? Those are things that we consume that our bodies don't make for us. All right. So glucocorticoids, I just want to touch on real briefly. Glucocorticoids are really cool. They're important for a bunch of different things like lung development, carbohydrate metabolism, but the one I really want to talk about real quick for just a minute is our inflammatory response. So there are two big reasons why you're gonna make a glucocorticoid hormone. Number one, you haven't eaten in a while, right? It's low blood sugar. So that's one thing. So you have low blood sugar. The second thing is you're stressed. So you're probably making some glucocorticoids right now, right? Right before your exam. I understand that. So this is why we think that when people get stressed out, they're more prone to viral infections, right? So if you make this glucocorticoid because you're stressed out, basically what it has is an anti-inflammatory effect. So if you're stressed out, it's going to try and calm down your immune system, which is a good thing, right? Except sometimes it'll calm it down too much, and then you don't have the appropriate immune response to a viral infection, right? So that's why we think that people get sick when they get stressed. But we can basically hijack this system, and we have. So what do you take if you have asthma? Albuterol, right? An inhaled steroid, right? An inhaled steroid is glucocorticoid. So it is actually going to bind to the same things, the same glucocorticoid receptors. And what's it going to induce? An anti-inflammatory effect. There are many different kinds of ligand-activated glucocorticoid receptors. And depending on what substrate you use as a ligand, it will activate a certain part of the anti-inflammatory response, right? So we can have our inhaled steroids. What happens if you have joint problems? What do they give you? An injection of a steroid, right? It's a different steroid, but it's going to have an anti-inflammatory effect. What happens if you have a skin rash? What do they give you? If you have a cut, you get Neosporin. That's true. But what if you have an allergic reaction? Well, Calman's over the counter, but a topical steroid, Grace, that's right, a topical steroid, right? Why? Because it's going to bind to the transcription factors that are inside of your cell and going to imitate an anti-inflammatory response. So this is what these drugs that I've just been telling you about are classified as
like cholesterol ligands.
They're not natural ligands, right? We design them in the lab.
Exactly.
So Adele's getting close. So they're drug therapeutics, they're inhibitors. What kind of inhibitor? Agonist or antagonist? If they mimic the same thing, right? The glucocorticoid receptor, when it binds its ligand, it has an anti-inflammatory effect. So if I put this drug in and I mimic an anti-inflammatory effect, what am I, an antagonist or an agonist? No, you're an antagonist. You're an antagonist. Because you're mimicking the same response as the ligand, right? You're like competing for that activity. Yeah? So why does it work? The question is, why does it work? these drugs have insane affinity for the transcription factors. Like if you called what naturally occurs in the cell like a 1x, right? You're going to go up to like 50x, 100x binding affinity for these drugs than the natural targets. And so you're going to get a massive and very quick response, right? How quickly when you do you know anybody who has asthma, how quickly does an inhaler work? Very fast, right? And that's really, really important. So that's how you can do that, because it binds as an inhibitor, actually, right? Okay. Oh. But I thought the antagonist induces the same signal. It binds in the same place that the normal signal binds. It induces the same response. So yes, your natural ligand cannot go in and activate that receptor, but it's not a covalent, it's not a covalent interaction. It'll pop out, right? And so eventually you'll go back to normal once your body metabolizes the drug. So an antagonist doesn't necessarily stop the signal. The agonist is going to stop the signal. The antagonist, like if somebody's bothering you, they're antagonizing you like crazy, right? It doesn't stop. The signal just keeps going. But an agonist, it's going to bind to the site and then you can't do anything. Does that make sense? It won't respond to the natural ligand because it's bound. Right, it's going to bind in the site, but it's not going to induce the downstream signaling effect. Yeah? Good? Maybe. All right, that is the end. That is the end of chapter 8. The end. Okay, so what I want to do is I want to go and pull up our chapter 8. Let's see, we did some of these. Which ones did we do? Here, I think we're picking up here, right? Okay, so I'm hoping this kind of helped you to organize in your mind the pathways of some of the really important ones that are going to be on the exam, right? Did you try these? And everybody checked in on attendance, right? Okay. All right. So PI3 kinase, right? This is one that's stimulated by insulin. So the insulin pathway, what's going to happen in the insulin pathway? Our insulin receptor is going to bind to insulin, leads to the activation of what? IRS, and IRS is D. Okay, so this one should have been D. IRS in turn activates PI3 kinase. Now I'm going to give you a hint. I'm going to tell you on the exam, every time that there is an abbreviation, I give you both the full word and the abbreviation. There are so many abbreviations that I don't want that to be something that trips you up. So I'm going to look over it again today, but I'm pretty sure that I got every single one, that it is all spelled out and abbreviated. So no matter which way you studied, you should be good. Okay, so PI3 kinase. PI3 kinase does what? It produces what? PIP3, right? We were PIP2 with our PI3 kinase, we get PIP3, right? So just add that phosphate group on. That's the definition of a kinase. So PIP3 is D. Okay. The PIP3 kinase is a dimer of 110 kilodalton catalytic subunit and a regulatory subunit containing SH2 domains. Binding of the PI3 kinase regulatory subunit to phosphotyrosines induces a conformational change of the catalytic subunit. right? While blank our association with our regulatory. What's going to happen to our association with our regulatory subunit? Is it going to maintain or disrupt? You got a 50-50 shot. It's going to maintain it. It's actually going to maintain our association with the regulatory subunit. But what changed is the catalytic subunit, right? That's where your conformational change made it. All right, so once we have our proper phosphoinositide is generated by PI3 kinase, now we have this lipid, right? This PIP3 that's got an anchor, it's got a lipid anchor into the membrane, right? Remember that? And so the lipid acts as an anchor for what? So you had to go in your book for this one. The plextrin domains, pH binding, right? Now, we didn't go quite this far into lecture, but you can go in the book for that, but I'm not going to go that far in the exam. But the PI3 kinase leads to increased glucose uptake. Does that make sense? If you're activating by insulin.
Yes, it does make sense.
Insulin is the I've eaten.
Now
Yum.
you get your glucose under control.
I need to get my glucose from my bloodstream into my cells. And so I'm going to increase glucose uptake. You need to know this. You need to know the hormones and what the downstream signaling effect is, like all the way at the bottom, what's going to happen, right? And glycogen synthesis to do what to our blood glucose levels? We're going to lower them. We're absolutely going to lower them. Okay. So I need you to know the beginning upstream things, right? Your book gets into some super detailed thing kind of down at the bottom. But I want you to know the beginning, and I want you to know what's the goal at the end.
So
right?
basically, you want to know like what starts this whole reaction, what ends. That's basically what
And what the end product is.
so the start of the reaction and the end product.
Yes. Like what if you're getting not insulin, but what if you're getting glucagon as a signaling molecule? How's it going to be different, right? You should have a complete flip-flop in result, right? Okay. All right, so our insulin signaling, right? So let's try to assign, here's our diagram. What's what? So this little pink thing is insulin, right? So this is our insulin receptor. Okay, so what happens? Our insulin binds, creates a conformational change in the beta. So these are our beta domains, right? This is a beta domain. The beta domains auto phosphorylate. So now that we have this phosphorylation, now what can bind?
Yeah.
Well, this first blue one, remember, is IRS. Why do we call it IRS? Insulin receptor substrate. Okay, so it's binding to our insulin receptor. So now that's kind of our bridging protein that becomes phosphorylated. Now when that protein becomes phosphorylated, what can it bind to? PI3K. PI3K can now become activated because it's bound to our phosphorylated IRS, and it is going to take PIP2 and turn it into PIP3. Okay, so how far did we get? We did something. So what was step number one? Step number one should have been insulin binding leads to tyrosine phosphorylation of these. So this should be D. So here's our order, right? Okay, I think we got to 2, right? So 2 should have been that PIP2 is phosphorylated by active PIP3, so that should have been B. Okay, so now we've got PIP3. What happens to PIP3? Now we have PDK1. PDK1, right? binds to our PIP3. And what else binds to PIP3? So this is another PIP3. Both of them are PIP3. AKT. AKT also binds, right? Only to that group that has a third phosphoryl group. Okay, so what do we say? So what is 3? 3 is, we did D, we did B, right? Okay, so then what's next?
Protein binds PIP3, right?
Proteins bind to PIP3, yeah, like PDK1 and AKT. Okay, so they do that through those pH domains. Now, once they're activated, PDK1 phosphorylates AKT, and then we go on this massive downstream kinase pathway that you don't have to know all the details of. Okay, so then 4 should have been C, right? Okay on that? I thought the picture might help you. All right, so now, how is this diagram different than the previous one? So we have really 2 pathways that get stimulated with insulin, right? This is the one we've already covered. You will not have the pictures on the test. It will just be words on the test. It will not be pictures on the test. But I thought the pictures might help you to kind of organize it and to study. You know, I don't know, they help me, so I don't know. All right, so we see on the right-hand side, those are the same pathways that we've taken before, right? But we have kind of an alternative pathway that happens as well. So what's going on here? Excuse me. So our insulin is still going to bind to the insulin receptor here. So what's going to happen first? So we have our peptide hormone insulin binds to the extracellular. So this is A, right? Number 6 is A. It's going to bind. Now what's going to happen after it binds? After it binds, then we're going to change the conformation of our beta subunits, right? So this is pretty similar. That's B, AB. Once that binding occurs, we're going to induce a conformational change through the molecule. Then what's our next step? Where does C go? What number? C goes to number 7 because this is autophosphorylation. Okay, so we have autophosphorylation of our tyrosine. Now, conformational changes in the beta subunit stimulate insulin receptor mediated tyrosine substrates. Okay, so we're going to phosphorylate tyrosine substrates. What are our tyrosine substrates? You could have gone either way with this. Where should D go? At number 4 or number 8? Number 8. Good, number 8. So this is IRS, right? And we know that IRS is phosphorylated by our tyrosine kinase receptor. Okay, now what happens? Is it one question for these order questions? Is it one question or a collection of questions? Well, so this one was just to kind of help you organize the steps. Mason, I'm not sure what the question is. You just kind of organize them, right? If you were going to go in order, okay, so what happens after IRS gets phosphorylated? This is kind of common. The phosphorylated IRS, now we're going to skip. The phosphorylated IRS substrate binds to SH2 domains on PI3K, excuse me, right? So this is PI3K. So that one is I. So we kind of jumped there. And so what's going to eventually happen? These are your PI3K signaling proteins, G signaling proteins. And what are they going to induce? Glucose uptake, right? Because we have high blood sugar at this point. Glycogen synthesis. That's how we store sugar. Okay, so that's cool. But think about this. When you have high blood sugar, that means you're eating well, right? What is your body then able to do if it's being fed? To grow, to make new cells, right? You don't want to do that if you're in like starvation mode. But if you're not in starvation mode, if you're being fed, we want something that is going to increase things like cell division. So a lot of times you have simultaneous pathways that are happening, right? So we know that insulin can trigger this pathway that controls blood sugar levels. But insulin also has that same, the same signaling molecule has a different pathway to induce changes in cell division. And this is a G protein dependent pathway. So what happens in our G protein dependent pathway? Look at number 4. What should go at number 4? We've phosphorylated our receptor tyrosine, our beta subunits, right? Now what happens after we phosphorylate our beta subunits? We have SHC, I could speak today, right? So this one has homology, a homology domain that will recognize that phosphorylated receptor. And it acts as an adapter protein, and it's going to activate what? The GRB2. GRB2 is an adapter protein that will adapt us to, what's the green one with the 2 on it? That is SOS, son of sevenless. Son of sevenless. Okay, so son of sevenless does what? Acts on RAS to activate RAS. So RAS becomes activated, which can then activate this whole kinase signaling cascade. So we call this the MAP kinase cascade. Okay, so we got to go back and we got to put letters to these numbers, right? So what's number 4? Number 4 should be, now we want to bind to our phosphorylated tyrosine receptor first. So that should be E. So 4 should be E, right? Now that we have that bound, that's our SHC, right? Now, 3 should be our phosphorylated SHC interacts with GRB2. So that's F. Then we have our GRB2 that's activated that can bind to SOS, right? So then SOS should be, SOS accompanies GRB2 as they translocate to the membrane by SH3 domains. So that should be G. And then the last one, where we have activation of RAS, should be here, is a GTPase exchange factor that activates RAS, leading to MAP kinase signaling. So that's H. Yeah? I'm telling you that on the exam, there are going to be some things that you have to order.
Okay.
Okay? Do we have to know? No, you do not need to know specifics of the MAP kinase pathway. You just have to know that you have this kinase cascade, and we call it the MAP cascade. I know your book goes into it. We're not going that deep. Okay. I think that's deep enough for us. Yeah. Okay. All right. That would be RAF? Yes. 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 Those are all the things you don't have to get into the details of. You just have to know that's the MAP kinase cascade. So you have to know that RAF's going to be that RAS is going to be activating all this downstream stuff, but you don't have to know what all those downstream stuff are and the order of them. And it's done by kinases, right? This is a pathway that is so dependent upon kinases. MAP is kind of the abbreviation for all of the downstream kinase signaling cascade. It's all about kinases, the MAP kinases. Make sense? So you don't have to know all those details. Okay. I'll look over these. Yeah? No? Some people, yes? All right. So a lot of times you have different signaling pathways, right? That are going to end up in different results, right? So sometimes one signaling pathway can activate a different receptor's signaling pathway, kind of like what we saw with that G protein being activated by an insulin receptor, right? We call that crosstalk, all right? So I'm going to pretend that you made a new drug. Congratulations, way to go. You made a new drug, right? And you now know that it activates cell growth. So this is a signal to your cells that they can start growing. Now, in order to start growing, right, we know that in our MAP kinase pathway, that ERK activation is going to happen. So this is a phosphorylation event on a kinase, right? So we know that that can lead to cell growth. I want to know, is the drug that you made activating through this ERK inside of the MAP kinase pathway? So is it dependent on the MAP kinase pathway? I can use an antibody, like a monoclonal antibody. Remember that? That only recognizes one epitope. And so that epitope, in this case, is a phosphorylated version of the ERK kinase. So we call it p-ERK. That's phosphorylated ERK, right? I'm going to find out that my drug blocks the phosphorylated ERK. So here's my normal cells right here. I have no compound. I have no inhibitors, nothing. I add my compound, and what builds up in the cell? Phosphorylated ERK. My total ERK levels haven't changed, but I went from having basically none to having all of the ERK phosphorylated. So I know that my protein of interest is inducing phosphorylation of a downstream kinase pathway, right? All right. I want to know which pathway my compound is working in. So what I'm going to do is I'm going to incubate it with some inhibitors. I'm going to incubate it with pKa inhibitor, and I'm going to see what happens. I'm going to incubate it with RAF inhibitor, and I'm going to see what happens. So what happens to my phosphorylated ERK if I put in an inhibitor for pKa or an inhibitor for RAF? Phosphorylated ERK is gone, right? Phosphorylated ERK is gone. So let's see what we can figure out on what's true and what's not. ERK phosphorylation by the addition of our compound is lost when RAF inhibitor is added. Is that true or false? That's true. Now let's see if they made the conclusion right, indicating that the drug signals through the epinephrine receptor. Is the epinephrine receptor, is RAF in the epinephrine receptor? Nope. 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 Not wrong pathway. Wrong pathway. Okay. Now, ERK phosphorylation is increased when compound X is added to the dish. True or false?
False.
ERK phosphorylation is increased when compound X is added. That's true, indicating the activation of ERK. It's a phosphorylation cascade pathway, right? So is this true or false? If you become phosphorylated, you're going to be active. True. True. It initiates that downstream phosphorylation cascade, so that's true. Compound X activates ERK through a pKa phosphorylation mediated event. Does pKa mediate this phosphorylation? How can you tell? If you inhibit pKa, if this is true, if statement C is true, if pKa doesn't work, then ERK won't be phosphorylated. Is that true? Yes, if I inhibit pKa, I no longer get phosphorylated ERK. So this is true. So it's dependent upon pKa. D, loss of activation of ERK when pKa inhibitor is added prior to compound X indicates, oh no, that the drug is activating, sorry y'all, via a GPCR. Sorry. So what do we think?
Loss of activation of pKa more than prior compound X indicates.
Is pKa involved in the GPCR pathway?
No.
It is. It is. It is. pKa is in the GPCR pathway. Yes. Yes. There are multiple GPCR pathways. pKa is involved in the GPCR pathway, right? This one, I would not give you something that hard on the test. What I wanted you to do with this one is to use your book and go and look at the information and try to figure things out. So you don't have to know all about ERK and RAF and that kind of stuff for the exam. But I want you to kind of think about how can you inhibit something? How can you identify, is this pathway working? Is this pathway not working? You know, that kind of thing. So sometimes these questions aren't all about, do you know the content knowledge? But can you start to pick it apart and figure out how the pathway is working? Okay? So it's, I mean, these questions are open book. They're not.
What now?
The questions, these questions are open book. Like you're at home. You're wherever. You can. No, not the exam. No, your exam questions will not be as hard as this. The point is to have class harder than the exam. Then the exam's easy. All right. So tasting involves many different cell signaling processes that ultimately generate nerve signals. I don't even know what time it is. Transduced by the membrane depolarization. 8 minutes. Sweet taste results in PIP2 hydrolysis. Oh, that should be a key there. Okay. While salty tastes allow sodium ions to directly alter the membrane potential. What can you deduce about the signaling mechanism for sweet and salty? So if you're going to allow sodium ions to directly alter the membrane potential, tell me what kind of a receptor this is. A ligand ion gated channel. Gated channel. Right? Okay, and tell me about PIP2 hydrolysis. It does split into DHE and IP3. And what else? What pathway is that involved in? In the phospholipase C. So that is GPCR, right? So let's look at our answers. That's what I kind of like to do. I like to look at the questions and see what kind of information I can pull out of them. So choose one or more. Sweet utilizes the GPCR signaling pathway activating phospholipase C. Absolutely. Sweet and salty signaling pathways use the same taste receptor molecule. No, we just said that. No, we just said that. It can't be that, right? Sweet ligands bind to and close sodium ion channels, which starts a membrane potential. They don't close the membrane. They don't close the channels, right? Remember what we said about these channels? That they're a lot like passive membrane channels, right? And so when you have a high concentration, they just go across. right? And that's what changes your membrane potential. Okay, let's see. Sodium ions directly enter the cells, indicating the signal is transduced by an ion channel. Yes. So that was like in section 1. That was from section 1. So it's kind of a. It depends on which taste you're talking about. For salty taste is different. There are different, depending on your tastings, what is going to happen. But it tells you here, right? Salty taste allows sodium ions to directly alter the membrane potential. So there are some tastings that result in ion closure. There are some. Okay, the last one that we'll get to. Okay, caffeine, everybody's favorite drug, right? Caffeine leads to vasoconstriction. Yeah, raises epinephrine levels and increases nervous signaling. A metabolite of caffeine blocks phosphodiesterase. What is the most likely impact of caffeine on cell signaling? When you talk about epinephrine, what kind of signaling pathway are we thinking here? GPCR. So epinephrine, you're thinking GPCR. Okay, tell me about phosphodiesterase. It converts cyclic AMP or cyclic GMP into AMP or GMP, right? So it turns it from the cyclic form to the straight chain. Okay, now let's see. Choose one. Phosphodiesterase leads to increased PIP2 breakdown. True or false? That's false. That's false. We just said that it works on cyclic AMP and cyclic GMP, so you could skip that one. The vascular effects of caffeine are long-term, indicating a steroid-enhanced activity. Do we have steroid?
It's not steroid. It's not, like I just said, the original point.
Right. Inhibition of phosphodiesterase results in an elevated cyclic AMP levels. Is that true? Yes, if you inhibit the phosphodiesterase, it's no longer going to convert cyclic AMP to AMP. All right, leading to, and PKA activation. Does cyclic AMP, cyclic GMP activate PKA? It does, it does. Leading to increased glycogen breakdown, increased phosphorylation of events controlling the physiology of caffeine addiction. That all sounds good to me. Okay, D, caffeine directly binds and activates the protein kinases. Caffeine is a what?
It's a stimulant.
But it's an extracellular ligand. Is an extracellular ligand going to directly bind to and activate a protein kinase? No. Right? Applying what's in those pathways, I know, is challenging. Question? I can, yeah. Yeah, I'll post it. Yeah, I'll I don't know, write on it and post it. Sound good? Yeah, I'll do it later today. I've got like appointments all day long, but I will get it in before like 3:30 today. All right, y'all okay? And hey, my suggestion to you is don't memorize formulas. Understand concepts, but don't memorize formulas. Please don't spend all of tonight memorizing formulas. Understand, what does Km mean? What does Vmax mean? But don't go memorize all those kinetics formulas. It's going to be conceptual. You're not going to plug into the concentration of the substrate and the concentration of the inhibitor, and you're not going to plug in numbers. Okay? Does that make sense? Questions on the chat? You okay? All right, good luck studying. If you have a question, send me an email. Yeah. I cannot move the test. I cannot move the test. Oh yeah, the sign up is on Moodle. You can go ahead and sign up. I know that y'all, a lot of people have another exam this week, but I cannot change the day of the exam. We still have information we have to cover, and we've spent a week on this chapter, and I've cut down quite a bit of what goes into this chapter. So I'm not changing the date on the exam. I'm sorry. So if you would like to take the exam in class, please do go ahead and sign up just to make sure that there are enough slots for everybody. But I don't see it being a problem. Even if you don't sign up, if you want to be in here, come on. There's probably room. Okay, any other questions? Okay. So the notes. So in the notes, agonist that mimic. Hold on. So if you go to, okay, I'll be here. So if you go to where? Here we go. Nope. Surveillance. Where was that? That's reversible. What's that in the chapter? Blocks the binding. Yeah. I wanted to tell you which slide that was on. Oh, yeah. Should be in 7.5. Slide 23. Let's see. Is it earlier than I thought? That's not slide. I'm looking at enzyme-mediated reactions on slide 25. Oh, slide 23. No, that's common catalytic mechanisms. Did I not print out? Is it 8.2? Oh, am I looking in the wrong one? It's too much. Let's see. Oh yeah, that's right. Okay. Agonists mimic it, right? So you're right. Slide 23. Y'all are right. Activate receptor signaling by mimicking the natural ligand. Did I? Oh, y'all are right. I got that. I did that reverse. Y'all are right. I did that reverse. The receptor agonist activate by mimicking the natural ligand and trigger the same thing. Antagonist block the binding of physiological without promoting the structural changes needed for signal transduction. So y'all are right. I think you may have switched them up in class. Yeah, that's what I'm saying. I think I did switch them up in class. So the glucocorticoid drugs, right? Agonist because it's stimulating the anti-inflammatory activity of the glucocorticoid transcription factors. Awesome. Okay, and I'll make that when I'm I'll post in the announcements when I have the key done for this, for the extra questions, and then I'll post that in there too. Thank y'all for saying that, for staying on. I appreciate it. Y'all good otherwise? Okay, if you have any questions, send me an email. I'm going to stop the share and stop the recording. Okay.